his conference COVID-19 and Food Animal Medicine. He's a doctor of veterinary medicine from the College of Veterinary Medicine from the University of Missouri and has a Bachelor of Science and Agriculture from the Truman State University. Currently, he's an assistant professor and a state specialist in small ruminants in Lincoln University of Missouri, where he's a member of the Agriculture Academic Planning Committee, assists in oversight of the university livestock, among other things. Also, he's an associate veterinarian in Green Hills Veterinary Clinic, Moberly, Missouri, where he provides client herd health sciences to improve animal health and productivity, and consults on management practices and administrative medical treatments for cow-calf, small ruminant, and equine clients. He has been a practitioner and clinic manager from Warrensburg Animal Hospital in Warrensburg, Missouri, and has a professional association with the American Veterinary Medical Association, the American Association of Small Ruminant Practitioners, the Missouri Veterinary Medical Association, among others. We'll leave you with Dr. Chris Bachman.
Thank you. Appreciate the introduction. And I want to thank you all for having me, allowing me to come and present and talk with you all today. Hopefully we'll cover a couple of things that are at least interesting, maybe informative, and we may find as we look at veterinary medicine and COVID-19, it's maybe more frustrating than we find even on the human side, just because we certainly have not had the time and resources put towards it in a disease, though, that's still so new. There's just a lot to know. So I will see if I can get a screen share going here, and we will move along. All right. So again, we're going to talk a little about really COVID-19 and maybe its impact in food animal medicine. And from my understanding, there's a fairly significant animal production area where you guys are located. And so hopefully it may bring some usefulness. Unfortunately, we may find that we're kind of limited again just by information on a topic that's just so new and different. Hopefully a couple of questions we'll be able to address, or at least feel like we leave with some idea about is, does it appear that livestock are important in participating in SARS coronavirus transmission or COVID-19? And as you've probably heard today, COVID-19 more so being the disease process that comes about as part of infection with SARS coronavirus. What evidence is there out there as far as infection in animals? And we could probably clear that one up now and just say not very much, really, in the big scheme of looking at information. And then talk maybe more about precautions or ideas, or what are some things to put in place to try to limit some of the spread, whether it's important or not. And a lot of those things, if we will take good precautions now, even if it doesn't have a huge effect for COVID, even if coronavirus does not become a big problem in large animal medicine, that we're probably going to make our operations better because we'll have practices in place to help limit disease issues. And so some kind of quick reviews for a little bit as we talk a little about immunology and viruses. And you guys have probably seen plenty about it today, so we won't spend a long time. But a virus, microscopic organism, DNA and RNA, not really considered to be living or alive. And they need another organism. They need an animal. They need a host. They need somebody to help them continue to replicate and reproduce. And occasionally, like I said, we will find some of these that aren't a big issue, and some can be very significant issues. And as we get into some of the more specifics listed here, if they're coded, what their capsids are they envelope viruses? Those may be things we find out later on as far as disinfecting and getting rid of. But for what we're worried about, again, we just have these DNA or RNA strands. In the terms of COVID-19 or SARS coronavirus, we know they're an RNA virus. And so the big question is, what does that do? How do we deal with that? And I think some immunology background is necessary. If we look at testing, how do we know if it's going to be a problem for these animals? How do we go about finding out the likelihood of it being a problem, or is it present, whether it's causing disease or whether it's not? And that's a lot going to be dependent upon our immune response. And so the immune response will have two different arms that'll play a part. One is our innate immune response. And the innate immune response is a very non-specific, I do this every time I respond to a disease idea. So if you're invaded, whether it's a bacteria, whether it's a virus, there's going to be a standard protocol of here's what we do. And the hallmarks of that we may see as those evidence of inflammation, heat, pain, swelling, redness. And it's listed as it comes with that cost because we may cause local damage, but the body's going to respond. But it does so in a very regimented and predictable way. It's not specialized. It doesn't necessarily provide a lot of specific response. And we'll compare that to the adaptive immune response, where we're being very specific to the invader that is present. And in that case, we're going to develop a response, a plan of action to get rid of this virus or this bacteria or whatever disease issue we're dealing with. And we're going to have specific responses, antibodies that will bind to that disease, cytotoxic T cells that will kill infected cells. We're going to regulate that disease very specifically. That takes a while to happen. And one of the reasons that we're seeing this as this pandemic issue is it's novel, it is new, it's not a disease anybody has seen before, there's no level of immunity, and so we have to go through the whole process. It can take 10 to 14 days to have a good adaptive immune response developed. It might take longer. The benefit should be that we have some memory and that we're prepared the next time that we deal with it much better. And so we see these in diseases all the time, but when we bring in a new disease, we can see a lot of these problems arise. And we see these occasionally as animal diseases change a lot, that when we get a very new virus or one that changes enough, or a bacteria that suddenly gets access, that they can become aggressive and cause problems. And we can see a high rate of morbidity, or the number of animals affected. And we may also get very high rates of death or adverse outcomes. And so, again, as we move through that, realizing those different phases will come along and how we'll hopefully use them, we also have to split that immune response in the other direction and talk about active immunity versus passive. The things we have been talking about are certainly part of the active response. The immune system is responding to a foreign organism, and it, again, takes time for that to become specific or adaptive or specialized to what the issue is. Conversely, we can consider passive immunity, and that's where we are given kind of a free amount of immunity. We didn't have to work for it. It's available immediately, but it usually only lasts for a short period of time. Colostrum, or first milk from the mother, is usually where we see that most specifically show up, or where we're most commonly going to see that in animals. They need to consume those antibodies in the first 24 hours of life. They are have a special ability during that time to actually take those antibodies in whole and use them for prevention against disease. After that period, they will be digested and broken down into components just like all other protein sources. We can see a few other examples, probably the plasma transfusions, blood transfusions. We most commonly look at probably in the horse world, at least where I have practiced. It is not uncommon to find horses that do not produce milk, often because we have some toxins in some of our grass or fescue that result in a mare not producing milk or colostrum. We can test for that, and as a result, we may give plasma. So we're going to give a high amount of these protective antibodies, and we're just going to give them IV so they're there and really improve often the outcome. Tetanus antitoxin may be one in a more production commercial system where we're raising animals more for meat products, maybe milk products, that we may use. So if we're concerned about a risk of tetanus from punctures or wounds or bites, we can give these injections. They help negate that toxin and provide protection. So again, when we look at coronavirus, probably passive immunity may not be a huge discussion or topic. if we find this virus becomes important in animals, passive immunity certainly may be very important. We know in people, there's been discussions about using blood transfusions. These antibodies have been part of it. Certainly on the animal side, I think we're a long way from that being a really significant portion, but we'll kind of see as time goes along. Just a few specifics listed on this page, and really just to kind of highlight that we know there are a number of coronaviruses that are in animals and have been in animals for a long period of time. We know that the SARS coronavirus is a beta coronavirus. We believe it probably came from horseshoe bats. Maybe not a big surprise. If we look at rodents and bats, they have the largest number of beta coronaviruses of any species out there. So if it was going to show up, maybe that's just where it was most likely to come from anyway. We know there are several coronaviruses, at least 4 that are fairly commonly associated with common colds in people. So they're not tremendously new. We know coronaviruses have been around for a long time, some that can move around, and they can be very different. We know that pigs, whether you're looking at porcine endemic diarrhea virus, which had a huge impact in the US about 5, 6, 8 years ago, as it come in and caused really high infant mortality rates or death rates. It was present in other parts of the world in the 70s and 80s. We know TGE, or transmissible gastroenteritis, was a big pig disease several years ago. For the most part, it's controlled, but will flare up once in a while. We can compare that to cats who have a coronavirus, and they can have 2 forms. One of those can be a fairly mild GI problem or enteric problem causing diarrhea. And then they can have the feline infectious peritonitis. So if you're a little worried about coronavirus and COVID and where it's going, don't read about coronaviruses in cats. I don't think it's a model that you're going to have to worry about, but the FIP shows some really weird things occasionally where it can cause these large granulomas that create some problems as an odd virus response. There is a bovine coronavirus. I find it may be interesting. There is a lot of information, or at least a few studies which said maybe it was an important part of respiratory disease in cattle. Bovine respiratory disease in the US is a huge, huge industry and issue. So there were some papers come out thinking maybe it was important. I'll be honest, in my 20 years of veterinary practice, I've never considered bovine coronavirus as a huge issue for me to deal with or to look for. So is it because we don't know enough about it? Is it because it's hard to track? Is it because it's maybe really not important? Sometimes those questions are tough to answer without the benefit of time or somebody really looking deep. And maybe a good time to mention it may depend on new technologies. As we find things like PCR, and we'll talk a lot about PCR and trying to diagnose these diseases, it's increasing our ability to find these new diseases, maybe problems, exponentially. If you're into animal nutrition, they are looking at ruminants now and really being able to find out what's the real components within a ruminant. We have all these hundreds of different types of microbes that live there that help them digest plant materials. And so their ability to start understanding what's going on is really starting to take off and is going to be explosive for probably the next several years based on new technologies. And so as we sometimes get concerned, this is a very serious virus. It's done a lot of stuff we haven't seen, but some of that may come back to us because we haven't been able to find these things before. So as new technologies emerge, we may well find some of these viruses and diseases have been here. They've been doing similar things to this, I have no doubt, for forever. But as we become more enabled, we're just maybe finding these things out, being able to follow and understand them a little bit better. Quick look, this page I took, I believe it came out after the SARS coronavirus 1 in that 2002, 2003 timeframe. There was a lot of study done at that time as a concern. That virus didn't have near the spread, obviously an impact, but showed a lot of potential, very similar to the virus we see now. And at that point, this page just showed one of several that showed different coronaviruses. I mentioned the porcine endemic diarrhea virus. Oops, as I went a little too far. The transmissible gastroenteritis, the feline coronaviruses, some of the different species they infected, and all the different sites and locations, most of them being upper respiratory and lung or enteric intestinal diseases, but certainly a wide variety. So again, coronaviruses have been doing some of these things for quite some time. Unfortunately, this one's now making a lot more trouble for us. A couple of theories, and again, a lot of people, a lot more knowledgeable, informative than I have, but the things I looked at say, where did it come from and why do we care? And a little of that is, the more we know about where it came from, what the host may have been, and usually there's an intermediate host or somebody that helped to move that from that bat into people, may give us a much better idea on a veterinary standpoint of what animals should we be concerned about seeing this virus become a problem. Put a picture of mink up. They've kind of stolen the headlines over the last maybe week or so, that we've seen the large outbreaks that are occurring, and they are showing some interesting maybe problems of where not only were they being infected by people, but they were potentially being able to then actually infect people. So we have a jump of species there. A lot of concern about them currently, and really I think the bottom line comes from, it looks like most professionals to say, there may be some concerning info, but let's don't get too worried too fast. There's a lot of things that we don't know, that we haven't had time to research yet. A lot of that initial data that come out did show that ferrets, cats, and some Syrian hamsters did appear to be good or likely models of disease. In other words, the disease did seem to be able to be studied well in experimental conditions. Certainly mink have emerged and are very similar to some of those. Others, such as the raccoon dog, has also been brought up to maybe be important. So we see a few of those species. We'll talk a little bit more later about some of the animal species that have been evaluated from a more livestock perspective. For the most part, many of them don't appear to be great. And I have down here on the bottom, you see other species, ducks, mice, pigs, dogs, do not appear to be good models. So it doesn't look like the virus does a great job of mimicking what we're seeing. And so those species, one, won't be good models, but two, may give us some assurances from a livestock perspective that they're not going to be huge concerns. So kind of what we know to this point is there's little to no evidence that domestic animals are easily infected. And we can probably maybe take mink out of there. Mink have kind of proven that wrong, so as we move them down the list. We see a lot of things written, some in a lot of news journals, about other animals being infected, and there have been some trials. Dogs just don't appear to be overly symptomatic. In some of these mink farms, they have evidence that those dogs may show evidence that they have been infected, but it does not appear to be a serious issue for most of them. Most of the reports we find also suggest dogs are not that affected. Cats, whether they be large cats or regular house cats, more susceptible than dogs. We see more of them positive. To date, the best I can find, most of them don't appear to have serious disease. We will see instances where those animals appear to even have two in the same household. One is affected, one is not. Most of them have been fairly mild. Cattle, I've got another report on that later where they've done a little bit, but really very limited concern of disease in most of these livestock species. So again, we have kind of, I think, little concern at this point. There is concern. Certainly we need more watching. And part of this is we really just haven't seen the resources put forth to really evaluate animals well. In my opinion, I think it'll be a couple of 3 years, so 2 to 3 years, before we may really know what those impacts were, how significant it could be. It's going to take some time, a lot of testing, a lot of man hours and labor to find out, was the disease present? Does it appear to be there in any amount? But it doesn't look like it's going to be a huge concern. When we have found this virus in any of these domestic animals, mostly related to cats, now we're talking mink as well, they've had very close contact with people that have been infected or had the disease. Most of the animal signs have been mild. Mink may be a little different. Most of the rest have been very mild if they've been clinical to any degree. Some don't show any evidence of disease. And we see that in people as well, which is just another complicating factor. And the big thing with most of these pets, so before we brought mink into it, is that most of these pets, and we'll look at a couple of questions in a minute, not only were their signs mild, but they didn't show evidence they were transmitting that virus, either to another animal or to people. And so we'll get to that a little more specifically in a minute. So in the US, the Center for Disease Control kind of gave out what we need to know. And this hasn't changed a lot, even over the last 2 to 3 months. Right now, we don't really know exactly where it came from. we think there's an intermediate host somewhere that may help this move, whether it developed in those species, became infective, and that's how it got into people, or did it get into people and it changed enough to become virulent or active and infective more so. But based on the data, you need really specific close contact situations for most animals to contract the disease, if they contract it at all, where their signs are mild. But again, there's really limited data. And if you want to look at scientific data, and we tend to rely on that a lot, it's just not going to be there for a while. There are a lot of anecdotal reports of, hey, people are running a few tests here and there. Good to know species we may need to look out for issues, but we have to be pretty careful we don't overinterpret a lot of that information either. So as we start to look at this, I think from the veterinary perspective, I think we can look at kind of 3 or 4 different steps or stages that we may ask questions about to find out kind of where we're at. And really the first question is, can it infect that animal species we're concerned with? Can it infect a cow? Can it infect a goat? Can we get it in sheep? Could it show up in pigs? We look at these livestock species we depend on for livelihood or food and sustenance. Is it going to be a problem in them? And in some of those, it doesn't appear that it's going to be really significant. The few that it shows some replication, maybe a few cows once in a while in one study, is that what does that mean in a very small study? And we haven't really seen a lot of disease, but they've given a very specific situation to let that disease occur. It didn't just naturally happen out in the world. And most of those times that disease occurred under very specific laboratory conditions where we really challenge that individual. If the animal can get it, the next step becomes, so does the animal get sick? Or is it one of those things that they can get? It may even replicate, it may go through a life stage, but they don't allow it to move on and go anywhere. So there may be what we call a dead end host. It shows up, maybe it lives there, but it can't really replicate, it can't move on. And maybe it doesn't even cause any disease. And as I mentioned, moving on, we see step 3 might be, okay, well, now they get the disease. Maybe it does cause a little illness, maybe it doesn't. Maybe more importantly, can that infected animal now infect another animal of the same species? So if a cat picks it up, can that cat give it to another cat that lives with it? More importantly, from the livestock perspective, we're going to worry, so if it gets in my herd or in my flock, is that a concern that's going to move through? And then the last part of that can be, okay, if it does all those, can it transmit to another species? That's what we've seen. It appears to do in mink. And the concern with that is, do we have this new reservoir? So it goes through, we get that adaptive immune response in people. We have a specific immunity to kind of fight it. But now that virus is living in another population. That gives it opportunity to change a little bit, and it lets it continue to circulate and be around so that we may have more exposure. And so there's several of those questions we may have to look at and try to answer and say, do these things occur? And again, it's probably going to take some time. And we'll kind of look at what test options do we have to try to see, are those things happening and do they occur or not? So again, sheep, goats, and livestock, at the time, the CDC, so Centers for Disease Control in the U.S., their latest outputs, which are probably August, and really no documented cases of significant issue. There have been reports I have read in different news outlets where people have found some evidence or antibody, or they picked up virus, but we just don't see that we're finding significant disease. And I think the important thing here is, if you're involved in a livestock production system, or if you're involved with animal raising, or you're working with people that do, when we see a new unique virus, we see it a lot like what we're seeing on the human side, is we expect to see a large number of animals affected. So if you wean calves today and you find some that have respiratory disease, that's not unheard of. We expect when we wean calves, depending on how we've prepared them for that, from vaccine standpoints, from stress, from management, that we may see a few get sick. And if you've done it for a while, let's say you have 5% of them that typically have a fairly big problem. If suddenly you wean calves and you're seeing 50% of them have a problem, now we're concerned that we need to be looking for something new and unique and different. We're still going to, at this point, think we've got a new bovine respiratory issue. We've had a disease we know about. And if we start ruling all those out and we can't find them, then that may suggest, is there something new or something that's crossed up? So looking at Do we see unusual disease presentations? Animals that are exhibiting signs at a large number? Again, because it's new, they have no adaptive immune response. Are we seeing unique evidence that more animals than we expect or should expect are showing signs of illness or some odd presentation? I put under new information, and I have a hyperlink at the end of this presentation. It's the same one. So the OIE came out with a new recommendation talking about farm biosecurity, a good summary of what's known so far as animals. Are they a high risk, a low risk, a medium risk? What's the risk of bringing in the disease? It's a very good handout. Again, there's a hyperlink at the end, but that's a good one to check out. So if you're in the ag sector, if you're just interested, I think it provides a very nice idea. But again, there's just a lot of unknown. As we look at future screening and potential testing, we'll talk a little bit about what some of those concerns could be. So, antibodies and exposure disease is the one we talk about a lot. They're fairly easy to measure. You can draw blood. You can get an antibody level. As we mentioned, one of the problems is the antibody doesn't necessarily tell you that much. You had to have the disease and you had to mount an immune response to have antibodies. And so if you have animals that are currently not been exposed, or animals that are currently sick but haven't had enough time to develop antibodies, or they're so minimally affected that their immune response is very low, you may never find antibodies. It is one way, though, we could screen and see, has the disease been here? Does it appear they've had some response? If you have antibodies to that disease, you have been infected, even if it didn't cause you a problem. Some other ideas we can look at are PCR, so real-time PCR. PCR looks for small bits of genetic material from that virus. It amplifies it, so it makes a bunch of copies so we can find it. And that certainly is what we look at with a lot of people in diagnosis, and that may be one of the ways we look at it. The problems on the animal side will be, if they're animals that don't show evidence of disease, we're going to have a really hard time testing them at the right point. So it could be they have the disease, but they don't show any signs. Maybe if they could transmit it, how are we going to know that? PCR is only going to be good if they're currently infected. It can also be a problem that if they are contaminated. So if you decide you need to test your cat because you felt bad and you want your cat tested, if the cat's been around you, maybe been licking on you, picks up the virus, we may get a false positive on that cat. Maybe we find pieces of the virus. The cat didn't really have it. It wasn't within the cat's cells. It wasn't replicating in cat cells, but we picked it up on the swab. Maybe we just handled it poorly. So there are some caveats as we go through these testing ideas and start looking for it. We got to make sure we, do them the right way and realize sometimes we got to interpret what that test is. If we're getting an odd positive, was it really there? Virus isolation, it's listed, and that's probably going to be a lot more research setting or where we've had disease investigation. We're going to try to grow that virus in cells in a laboratory, and that lets us do things like sequence it. How similar is it to another case we've seen? But it's very slow. It can take a really long time. It's not going to be applicable. The average veterinarian, the average producer, probably not going to be involved in that unless you're in a very unique situation. Most of the time, if you're involved, you're probably going to have PCR ran. Hey, I've got some odd sick animals. We're going to run a PCR to try to look for evidence of a disease. Or probably more commonly, we're going to screen for antibodies. Was there evidence this disease was here at some time? Doesn't tell us how bad the disease was, doesn't tell us how long it lasted, just says that it was here. And again, it brings up, we talked in immunity about that cell-mediated side, the T cell side. There is no great way to evaluate that. It's super important. It's really important as far as how we expect the immune system to work, yet it's really hard to evaluate. So again, we have ways to look at it. Certainly none of them are perfect. Potentially the biggest thing I think we may get out of all of these things is, again, at this time, there's not a lot of evidence to say that coronavirus is a huge concern for our livestock species. But it probably really does highlight and bring to light that if you don't have a biosecurity plan, if you don't have a plan of how to keep disease and issues out, we probably really need one. That we're going to continue to see new challenges to animal health, either because we have the technology to find them or because they're just emerging. There's more interaction. But a good biosecurity plan probably pinpoints that we need to have these now if you don't. And have a plan set up of how do we prevent disease being brought in? How do we deal with it if it's here? How do we bring in new animals? A lot of what I think we'll look at for biosecurity as far as coronavirus is concerned, at least in the short period, probably for the next few months to next year or so, is how do we prevent it coming in just so it doesn't have a workforce or a work staff implication so that we can keep the continuity of operation. So if you're on a fairly large farming operation, there's a certain amount of care that has to be done, especially when we get into dairy areas. How do we keep people free so they can keep working? If we don't have somebody to milk cows, milk goats, and process them, that's a huge challenge, presents a lot of health problems. There's just a lot of issues. I think we'll see more applications of biosecurity and just keeping the workforce healthy than we're going to worry about on the animal side. But if we do a good job on the workforce side, hopefully we're limiting any odd exposures that we could bring in as well on the animal side. In the U.S., I'll point this out, we have a new program for sheep. It's a little behind some other species. We are considered a minor species in the U.S., sheep and goats both, meaning we just don't get a lot of attention comparatively, especially with beef, pork, and poultry. But we have a number of plans for all species now called the Secure Sheep and Wool Program or whatever the other species. And it's really just centered around developing good biosecurity protocols. The real push for this is if we see foreign animal diseases, things like foot and mouth disease that will enter, and we'll see much what we've seen in the human pandemic, this disease that just runs rampant and can cause huge amounts of problems. So they have a great resource. Again, the OIE, World Health Organization, has come out with some new recommendations. And I think, again, if you're working with operations or if you're looking for things that may be, I think, very important in animal science, animal production in the future, is good understanding and implementation of biosecurity is going to be something that will only increase as we move forward. And this was kind of a summary of that report, and I think it was dated today, but they come out from the Animal World Health Organization, or OIE. Those are guidance on working with farmed animal species. And a lot of this was driven from the mink industry because they've had such a big issue. Mink are unique to this point that they not only appear to get the disease from people, but maybe they can pass it back. Keeping in mind our livestock species do not appear to be near as susceptible, but we still get a pretty good idea of how to make some preparations. What they listed is the risk assessment for cattle was very low. I think they had evidence of one study that had been done, and they managed to make a few animals seroconvert in a very laboratory type setting. So they considered them to be very low, negligible animal to human transmission, and negligible for transmission between animals. So from that side, we're really just not terribly concerned at this point. Poultry, they had no studies showing any evidence of transmission that was occurring. So again, negligible risk in any movement on that side. And pigs were assessed to be very low. Pigs don't appear to be a good model. That may contrast a little bit with what we've seen with other viruses. We've had a few outbreaks of influenza issues where we've seen some transmission between pigs and people. But when we've seen that in the U.S., it's usually been in context of fairs. We've had people in very close contact with these animals. They're usually show animals. They're with them all the time. They're laying on them, vice versa. So again, it's not a huge issue. It's usually very isolated. And again, it appears to be really low in those livestock species for us at this time. I think the other big thing then we have to try to assure people is when we talk about is what's the safety of our products. And for a lot of what I deal with, that becomes about meat product. Certainly dairy and milk products are also part of that. And we have a certain amount of that that's also maybe those companion animals, whether they're just pets and buddies. We have a fair number of people where I'm at whose goats are really more of a pet and they just like to hang out with them. But we may see some unusual close contact. They kind of snuggle up. We do yoga with goats now. So you may get some unique contacts, but what is our risk? We have some working animals, guardian animals, which can be very important in the sheep and goat industry here, preventing predators and wildlife issues. I think the big takeaway from the meat safety standpoint is we don't see any evidence this virus replicates in animals. And so therefore, it's really going to be more of a contaminant and a surface issue with most of our food meat sources. As we convert that muscle into meat, we get a lot of changes. Cells lose that homeostatic ability. We get a big drop in pH from about 7.4 to 5.4. So even if they were in a few of those animals, can they survive that? The food appears to be not a big source of concern. And really, if you're using good food handling, I think your meat product should be fine. It's, again, been more of an issue where it's just the people producing that. So those facilities that are producing it, we're losing a workforce shortage. Milk and dairy should be fairly similar. Again, we're not seeing these animals become infected that we expect that virus to be within the product. It's more of a contaminant. If we're using good food practices, most of those foods should be pretty darn safe and really don't bring a big concern. Raw milk, maybe there's some differences there. I'm not a big raw milk proponent myself. Pasteurization, I think, saves a lot of problems. We can look at a number of diseases, some things like listeria, tuberculosis, mycobacterium tuberculosis, a number of things could be in there that if we're treating that milk pasteurizing, I think it just limits a lot of risk. But again, good food practices, I think, make those products really safe. I don't think that's a huge issue. Our big issue is keeping the workforce in place, keeping those farmers and farm employees being able to come to work and get that job done has really more been the focus of food concerns. And again, when we look at like meat products, we don't have living cells, and viruses infect living cells to replicate. So they're not going to infect meat because they're not a living cell any longer. And so again, that risk really becomes more of a contaminant where good food handling should provide plenty of safety for those products. And again, as I went a little far here, but I showed this hyperlink down here where the OIE shared some of those ideas. If you're looking at biosecurity, if you're concerned about different species susceptibilities, they have a pretty nice handout. Can certainly find you a lot more data at that point. So I think that's about all the excitement I had. So if anybody has any questions, feel free and I'll do my best.
Thank you very much, Doctor, for your valuable participation. I'm going to expect some questions that were made through the Slido platform. Just wait.
Sure.
Do you think CRISPR-Cas9 can be a useful tool to fight the possible infection of COVID in livestock?
I'm sorry, can you repeat the first part of that?
Yes, sure. Do you think that CRISPR-Cas9
okay,
can be a useful tool to fight the possible infection of COVID in livestock?
Well, you're probably a ways out of my specialty as you're getting into looking at some of those advanced techniques, and those things certainly could play into some part. I think they're just a few steps away to get that specific. I think right now we have some areas that, and maybe they are just as easy. I think at this point where we see such limited effects of animals anyway, we don't see a lot of clinical disease. How far are we going to push to start looking at these more advanced opportunities to look and find genes and codes and things like that? They may have a point. I think PCR and antibody surveys are certainly where it's going to start, though. Those are going to be the easy to run, where we're really going to pick them up and probably put more of these advanced testings in as we start to really try, if we find a problem. So if we use some of these, how we're going to go in and find that. But again, you get over my head pretty quick with those, and somebody may be able to apply that, make it a lot more applicable than I'll understand. So.
Okay, thank you. Other question is, how can we prevent future outbreaks of novel zoonotic diseases?
Well, that's a great question. And I don't know that we can really necessarily say we're going to prevent them. If you look at trying to prevent these unique disease outbreaks, a lot of it has to do with that interaction level. And there's a certain amount of that that's just going to happen. I suspect we have seen some of these zoonotic diseases. We know there's some that emerged over time. They have all come from somewhere. And so it's maybe just a natural part of these disease evolutions. If we look at how can we kind of maybe have some effect, I think really it comes to biosecurity being a big part of that, because it's what we do to keep out unique diseases, how we monitor our group. Do we put practices in place at critical points? So if you're in food safety line, they talk a lot about HACCP or hazard analysis, critical control points. And if you look at this OIE document that came out, a lot of it's about what are these critical steps of where these interactions can take place to see disease. And if we put some of those in place, that we may limit some of those, I think that's how we maybe get ahead of it. I think there's a certain amount of just biology and diversity that's going to occur, that we're going to see these outbreaks. But if we can identify them early, we see unusual presentations, high numbers of animals that are sick. If we have good biosecurity where we can immediately shut down some of this communication and spread, I think that gives us our best opportunity. And then probably a lot of younger people that get a lot more of this genetic programming ideas that I do about what we may be able to do from a gene therapy or genetic. There's probably tons of options maybe we don't even know about yet. But for what we have now, I think good biosecurity gives us the best opportunity to find and then address and limit spread. And I think that's where my focus at least would be at this point.
Okay, thank you. Another one is, even if there's no evidence of animal transmitted the COVID-19, there are still a possibility that pets or cattle may transmit it to us?
I mean, I think that could be found. Doesn't appear to be at this point. But again, I think we're just too early on to say that it can't happen. And part of that is we're going to have time to look for it. Right now, at least I know where I'm at, the resources still are really focused on just trying to test people. The backlog of being able to keep up with some of those tests or who they've had communication with is really big. And so resources at this point just aren't there. I think if we see this slow down to some degree, we see more efforts put into that, we'll have a better idea. Again, it wouldn't surprise me if it's at least a year or two. I think it could be 5 years before we may really know, are they going to be a significant problem? I wouldn't rule any of them out. If I'm a farm manager at this point, I'm not overly concerned about my animals becoming infected, but I'm going to put some things in place to be safe anyway. I'm going to take good practices, keep me and my workers safe, and in doing so, I should really also prevent a lot of entry of disease for them. So I'm going to play it safe. Again, I think we're a few years away from really knowing that we can say, yes, it is or isn't. And then it could change at that point and mutate or become something new. But
okay, thank you. And another one is, did you see a problem that actual massive livestock activities create genetically similar animals in which new diseases have an easy environment to proliferate?
I'm sorry, one more time. I'm getting old and I can't hear very well.
No, it's okay. Do you see a problem that actual massive livestock activities create genetically similar animals in which new diseases have an easy environment to proliferate?
I don't know that I think that's a huge issue, but I think it always has to be a discussion, could it become? And if I understand that right, but where we're having all these animals that are very similar, it's really easy, I think, and I think we see it in the U.S. a lot when we get into beef producers. And I'll be honest, in Missouri, we're the number, I think, 2 or 3, and it fluctuates, cow-calf state in the country. We're not a real big state, but we grow a lot of cow-calf pairs. We don't feed a lot out. We're not big in total numbers, but in cow-calf operations. So we have a lot of small operations. And we tend to lean pretty heavily towards having registered stock, I think, compared to a lot of the rest of it. And it's easy to get caught up that we find maybe that 1 or 2 great animals, or we look at a lot of numbers and we say, and this is the way to go. And I think we do start to narrow our pool of diversity a lot. And one of the great parts of the people ask us, so why do we have so many breeds? And if you go on and look at the number of breeds of animals, it's just mind-boggling. It's hard to keep track of. There's so many. And the question is why? And one of the great opportunities there is because we preserve this genetic diversity. So if we find this particular type of animal is overly susceptible, there may be a breed out there that solves that problem. Similarly, we look at small ruminants more where I focus. Parasites are the bane of existence where we're at. They're a huge problem for a lot of people. With diversity, we find animals that can handle that. And so, yeah, I think we can with some of these large productions. Sometimes we zero so much in on great performance that we could lose some of that diversity. Hopefully, as a collective worldwide, though, we always have some of that diversity held on to try to prevent some of those big losses.
Thank you. Another question is, how do you think the coronavirus will affect the beef industry?
Well, I probably could do a lot well in life, or could probably be pretty popular if I could figure that out. And there's a lot of people know a lot better than I do. The big impact we have seen here has been from a backup or processing and what it's had. I think for the main part, that's what we've seen. I think for a certain degree, that's where it's going to continue at this point. It's really going to be if we can keep up with production, we can keep those people processing the product to keep it available, healthy, and keep those systems in check. I don't know that we're going to see huge impacts from it. There's a lot of discussion what's going to happen from an economic and a business standpoint, at least in our country, and what that response is going to be. I think those will be the things that drive that beef industry a lot at this point. Will we see other changes just as a result? Maybe. Maybe we'll see some upticks in biosecurity, how we go about that. I think we're going to see some research and testing. Overall, in the short term, I think it's really just dependent upon its effects on economy and consumption. I think it's going to be fairly limited outside of that at this time.
Okay. Another question is, I heard that the virus is fake and the government is inventing it to control us. I believe it. Do you believe it?
I think it's a real virus. I think it's out there. I think it's causing problems. I think we've seen too many people that have come up with it and are showing some signs and problems. Where it came from, somebody a lot smarter than me will have to figure that out. I think it's a real virus. I think it's out there. On the upside, I really think from our animal perspective, as a veterinarian, from patients I'm going to see or producers I'm going to talk to, I'm not terribly concerned it's going to have huge impacts on those animals at this time. It's, again, I think right now a lot more about how to deal with the human aspect of it. And again, who knows? Somebody a lot smarter than me will have to figure all those things out.
Okay. Another question, what is your opinion on the minks infected with coronavirus with a mutation and the danger of it?
And so I'm going to kind of resort to, unfortunately, what the really smart people tell you. And at this point, I don't know that it worries me that much. So there were reports coming out that maybe we needed to back off some of these concerns about vaccine and treatment and how is it going to affect those. And I think that's a real concern. But at the same time, we know viruses mutate and change all the time. If they didn't, they just wouldn't be relevant. The flu vaccine or the flu influenza virus is probably the great standard example. It's always a little different. It can change. It can mutate. Thus, your vaccines aren't always effective. They can be a little different. And it's why it finds maybe a new population that maybe I develop a great immunity to it, but it changes just enough to come back to be an issue. There's just as much probably also to do with that as what is my current state of health. If I'm really stressed because I was prepping for this, and a little worried about what to say, and I didn't eat well and sleep well, maybe I just get susceptible anyway. My immune system did not do a good job. But those viruses change and mutate. That is part of what keeps them going. We know these things are going to change a little bit when they move into new species. Maybe that gives them more opportunity. So I think it's kind of to be expected. We know they're going to do that. I think to be overly concerned about what it's going to do out there is not where I would be at this time. Is it something we need to watch for that next big change? Sure, needs to have an eye kept to it. We hope we make the best decision on how to deal with it. It's not a huge concern, at least for me at this point, but that would be my take at this point.
Okay, another question that I really don't understand, but I want to read it. Is, has PCR presented any similarities in the piece of RNA between animals and the RNA that PCR? No, it's because I don't understand the question. So I'm gonna ask to do it again, please.
Sure.
Because it doesn't make sense, the question. So it's just asking if they can do it again. So
okay. And then I said, and if we're looking at similarities of RNA or viruses in different types.
Yes, I'm going to read it, but has PCR presented any similarities in the piece of RNA between animals? And is the RNA that PCR looks for the cells the same? So I don't know what they mean.
Okay. Well, and I said, we'll see if they bring it up in different, but and how we go with that. I mean, I think certainly there's, some options to look at there where it's looking at RNA and what it's trying to find and what PCR does for it. And there is some question that we'll mention that when they look at RNA, is it finding whole segments or pieces? So as we amplify these things, there are certain tests, and again, it's getting a little over my head as you get into the very specific molecular genetics and biology of it all, but some of that PCR, if we use a quantitative PCR test, it could actually probably differentiate if we're finding just pieces or if we're finding whole sequences of it. But
okay, I think that was the last question.
Okay.
We appreciate your time, the information shared with us. Now I will present your recognition that is going to be sent to your email.
Okay. Well, again, I thank you all very much. I hope maybe it provided you a little something to think about. And, if anybody has questions or wants to reach out or anything, certainly welcome that. And again, appreciate you all for letting me present. Thank you.
Thank you. Now I'm going to read the recognition. It's El Tecnológico de Monterrey Campus La Laguna and the Bioengineering Student Society grants the recognition to Dr. Christopher Bachman for your participation in Encuentro Biotech 2020. Thank you very much, Doctor.
Thank you all. You all have a good evening.
You too.